Introduction there. I think this is a really important and timely topic, so I'm excited to just jump right into things today. So before I begin, I'm excited also to introduce Syntica. We're a global life sciences partner that's dedicated to advancing preclinical research. Our mission really is to support scientists by offering high quality equipment. We partner with over 20 different manufacturers that are located globally, and we act as a distributor and local service provider by performing the installations, the trainings, the post-sales support, and application. Ultimately, we're here to help you ensure that you're getting the most value out of your investment. So again, the Syndica team works alongside you to help educate, troubleshoot, and support just about every step of your research journey. So we offer a really wide variety of preclinical imaging solutions, ranging from ultrasound, MRI, to X-ray, PET, CT, and more recently SPECT. For the purposes of today's presentation, we're going to focus on how optical imaging helps to drive ethical animal research in alignment with new approach methodologies, a topic that I'll define here momentarily. So by way of session objectives for today's webinar, I'll begin by setting the stage for what's happening in biomedical research, which is this major shift towards what we call new approach methodologies, or NAMs for short. We'll learn about NAMs and see how they're complemented and supported by what's known and recognized in the preclinical field as the three Rs. We'll talk about how optical imaging as a longitudinal modality facilitates the movement of NAMs towards more mainstream research studies, and explore how the Newton 7.0, so our optical imaging system at Syndica, how it's designed to champion these ethical principles that are focused on reducing and refining animal testing. So then, of course, if we have time towards the end of the presentation, we'll save some few moments for a Q&A period. So I'd like to begin our discussion by walking us through the drug discovery process to gain an appreciation for the sequence of events that takes place before a therapy can be approved and become available to patients. First and foremost, a successful therapeutic product is one that addresses an unmet clinical need, meaning that a disease or condition is observed in human clinical populations, and then this becomes the catalyst for the journey that's going to take well over a decade to complete and cost billions of dollars before reaching the final stage of regulatory approval. So of course, up top there, starting at number one, that's our observation of disease in humans. Then we move on to the R&D phase, which represents more of a reductionist approach. This stage can take several years, involving viable target identification, high throughput screening, looking at different compounds, identifying leads, and then pursuing those into the next stage. Preclinical studies, these involve in vitro work and in vivo research solutions, where animal models have served really as the cornerstone for safety and efficacy testing for many decades now. Historically, we've seen an array of different species being used, kind of all the way from C. elegans, or roundworms, zebrafish, rodents, all the way up to domesticated animals, non-human primates, etc. They've really played a critical role in drug development, helping us to understand disease mechanisms, evaluate toxicity, PK/PD, as well as inform, our decisions about advancing therapies into humans. So really, in essence, we use animals in cell culture to reproduce and then reconstruct the disease that we've observed in humans, and then we test therapies here to see if they're safe and able to work as potentially a cure going forward. However, it's worth noting that many compounds are ruled out after preclinical phase before they advance into clinical trials. Those that do, once again, are assessed here for safety before we can even begin tackling the question of whether they're effective. So observation and intervention are therefore evaluated through these phases of human clinical trials. And again, this takes many years and many dollars to complete. Finally, we have the review and approval stage, where companies will submit a new drug application to receive approval by the FDA or Health Canada or other governing body. The monitoring phase continues thereafter for several years. So collectively, each stage here represents really a go, no-go decision checkpoint. And something that we'll get into on the next slide here really is that, of the thousands of drug candidates that are first identified, many are ruled out throughout this phase. So around 90% of drugs that enter a clinical trial never actually reach FDA approval, with about 30% of those being due to off-target effects or safety issues that arise. Meanwhile, there's the remaining 60 that fail because the therapies just didn't translate well enough from animals to humans. Now, it doesn't appear to be a matter of not pouring enough funding into supporting good science, but rather the fundamental fact that animal biology is different from humans. So, of course, with these, there are species-specific differences in physiology and immune systems. Some inbred strains of mice have higher homogeneity in their genotypes. There are challenges with fully replicating complex human diseases using these reductionist approaches. And even environmental factors like stressing lab animals, this can result in altered outcomes and then therefore impact translatability. Traditionally, these studies, they can take again several years to complete and use a large number of animals, all carrying the high probability of failing to predict human immune responses. And this means the challenge that we face in is scientific, it's translational, and therefore it's ethical. So the question becomes, how do we generate data that is both scientifically rigorous and more human relevant? What's being done to facilitate better translation of drug therapies from benchtop to bedside? And so what's great is that we have seen improvements in more recent years thanks to better cell therapies, biologics, and notably refined, improved, and more relevant animal models, as well as changes to the FDA approval process, which we'll get into in just a moment. So related to this, there is this increasing move towards new approach methodologies or NAMs, as well as human-centric models such as organoids, organ-on-a-chip technologies, and computational modeling, etc. NAMs, they offer a tool to assess the safety, efficacy, and pharmacology of drugs and therapeutics. And so these changes, they mark a pivotal shift from what's been required historically for investigational new drug applications, providing faster and more accurate human risk assessments and improving, again, the overall translatability of drug testing. Importantly, animal models, particularly genetically altered and humanized mice, will remain essential for understanding complex biological systems. So, of course, people have the question, are animal models going away anytime soon? The answer really, realistically, is no. We're far away from NAMs fully replacing animal testing, but definitely a more refined, more ethical approach to leveraging in vivo models is required in order to complement NAMs and again, improve the translational success of new drug therapies into humans. Of course, we'll dive into this a little bit more in the coming slides. So where did this concept then of NAMs kind of come from? So in 2007, the European Union, they established their REACH regulations, which is a framework that prioritizes reducing animal testing for hazard and toxicity assessments. In I think it was 13, came the EU cosmetic ban, which prevented testing cosmetic components on animals, forcing this industry-wide adoption of in vitro models. And then from there, we've seen a steady decline in the total number of animal procedures, while the use of genetically altered animals and humanized mouse models has actually risen. So in more recent years, the FDA Modernization Act eliminated the requirement for animal testing in certain drug approvals. And then last year in 2025, there was a harmonized joint workshop between the FDA and the NIH to formally establish that INDs must prioritize the use of new approach methods. And then, in addition, whenever animal models are required, researchers must justify how they are more human relevant, demonstrating again one's commitment to the three R's. So again, we are still far away from NAMs being able to fully replace animal models, but until then, efforts should be made to create more refined experimental procedures with the three R's in mind. So to briefly touch on the different types of NAMs, we have in vitro human-derived systems, which can include in vitro iPSCs or patient-specific models of disease, organoids, organ-on-a-chip technologies, these so-called microphysiological systems, 3D bioprinting, essentially to recreate organ-level function, allowing us to observe real human responses, but in a more controlled environment. We also have in silico tools and computational modeling, including artificial intelligence, machine learning, predictive models. bioinformatics, and off-target screenings, as well as physiologically based pharmacokinetic models. These tools help to understand drug distribution, toxicity risks, and immunogenicity in a fast and more cost-effective way. Third, we have this other sort of innovative platform category, such as ex vivo human tissues, high throughput cell-based screenings, microdosing and imaging in human volunteers. And on the top right, I've highlighted here that we have another sort of subcategory called refined enabling innovations, or REIs. So these are methods for contributing to the three R's, effectively benefiting both science and welfare of the animals. So this umbrella term really includes behavioral and technological advances that reduce animal stress and improve the quality and interpretability of in vivo data, contributing primarily to, again, the refinement and sometimes reduction of animal use. So now where NAMs and the REIs intersect, we're going to see better human relevance of therapies resulting in improved predictive accuracy. We'll see faster results, meaning that long duration animal studies are going to be less needed. We'll also see a cost reduction because animal studies and repeat experiments are very expensive, so less of them means lower costs. And of course, reducing the reliance on animal testing represents a big advancement in ethics and welfare. So collectively, these regulatory agencies envision a future where NAMs provide sort of the default evidence, and then refined animal studies are going to be used more so whenever they're absolutely necessary. Again, collectively, this all translates to better decisions at each of those different checkpoints in that drug decision or drug discovery process, fewer sort of late stage failures, and most importantly, safer therapies reaching patients faster. So really the call to action whenever it comes to adopting NAMs and refined in vivo methods is A, knowledge. So educating researchers on NAMs, offering trainings and workshops, and sharing success stories. Two is going to be funding, so increasing NAMs research and maintaining incentives for doing so. Three is encouraging collaboration by way of joint research projects and sharing databases and resources. And then, of course, number four on the right, which is going to be developing standardized NAMS protocols and a means for assessing their safety, reproducibility, and efficacy through the approval pipeline. So with NAMS being gradually more and more standardized, we're going to see or we're going to need rather a way to bridge the gap in biomedical research, and therefore leveraging refined in vivo methods that provide high quality human relevant data and championing these ethical and effective use of animal testings. Technologies such as optical imaging can become really important. So I think this might actually be an okay time for doing a pause for the audience poll. Gabe, I'm going to hand over the controls to you. And then you can let me know maybe whenever we're done, and then I'll continue with the rest of the presentation.
Perfect. Thank you, Jade. So before I launch the poll, Brita, I see that you submitted a question in the Q&A, and the answer is yes. I will provide you with your request after the session finishes. So for now, we will just take a brief moment to launch this quick poll question. Just give me a second while I launch it. There we go. So the questions are the following. What kind of modality do you use in your current studies? With the options being MRI, PET, in vivo micro CT, ex vivo micro CT, ultrasound, BLI, FLI, SPECT, DEXA, or other. The next question is going to be, which non-animal alternative is your lab currently using or planning to integrate in the future for biological research? The options being organs on chip, organoids, bioprinting. The next option is in silico and computational modeling, in vitro assays, and not currently pursuing NAMs directly, but are committed to the three R's of animal research. So while we take a moment to let everyone complete the poll, I want to share a quick announcement. Syntheca will be attending the Preclinical Imaging Consortium, where my colleague Owen McMillan will be presenting on the power and versatility of the Newton 7.0 optical imaging system. And this presentation will take place on April 14th at 11:06 a.m. We will also be at the American Association for Cancer Research from April 19th to the 22nd. And if you are attending, I highly encourage you to stop by booth 2104, which will be the Syndica booth, where we will be having the Newton 7.0 system, and it will be on display for you to explore and interact with. Be sure to save your dates, April 14th, 2026, for the Preclinical Imaging Consortium, and April 19th to the 22nd for the American Association for Cancer Research annual meeting. Okay, so I see majority of the attendees have answered the questions. Just going to wait a couple seconds just to give everyone a chance to answer, and we're going to move forward with our presentation. Okay, thank you for answering the poll questions. Jade, I'll give you back the presentation, and thank you so far for the presentation.
Awesome. Okay, so moving forward, now I've mentioned this concept a few times now, the three R's. So I just want to take a moment to introduce them and define them. So historically speaking, the three R's have long guided regulatory and ethical thinking in preclinical testing, and now really provide a foundation for understanding why new approach methodologies are central to the industry's future. So this framework was introduced back in 1959 by Russell and Birch, and includes concepts that, again, to this day, remain core aspects of ethical scientific integrity for the use of in animal research. So the first one there, the first box, we've got reduction, which means fewer animals without compromising your results. So that's powering your studies properly while not overusing animals, performing longitudinal techniques or imaging where, animals can serve as their own controls. And then, of course, sharing databases and preclinical repositories to limit the use of more animals. And the second one, so we've got refinement, which ultimately leads to better welfare and therefore better, more scientific, accurate outcomes. It's key for minimizing pain and distress and preventing unnecessary procedures using humane endpoints and relying on non-invasive imaging technologies, again, such as optical imaging, where you can achieve longitudinal and highly sensitive biological data. An important consideration as well is the implementation of precise models such as humanized mice to improve, again, the relevance and translatability. Finally, we have the replacement, leveraging alternatives to mammals, including non-vertebrates, in vitro assays, as well as in silico modeling. So under the category of REIs is the refined use of non-invasive in vivo technologies, which includes optical imaging. This longitudinal modality is highly valuable for monitoring biological processes in real time, including at the level of the tissue, cells, and molecular signaling events. It's really well suited for a range of different applications. So, tumor growth and targeting, immunotherapeutic development, biodistribution studies, you name it, the applications are quite vast. It allows us to visualize and capture dynamic processes within a living organism, offering again the ability to characterize more so the pathophysiological sequelae of events as they occur without requiring terminal endpoints for different tissue collections. Next, so in order for a biomarker or for an assay to be valuable, it must be highly sensitive and specific. I'll use standard bioluminescence imaging as an example here. So it fits these requirements really well. So because there's no excitation light source and instead leverages chemistry, there's an enzymatic reaction. So for example, we have luciferase, and then we introduce a substrate, so luciferin in this case, and then this results in light being emitted only by the cells that would be expressing the luciferase. So this is captured by the detector inside a fully enclosed and sealed darkroom, and therefore you're not getting the autofluorescence or any noise in the background. In that case, becomes very sensitive and specific. Now, with optical, we can also achieve a reduction in cohort sizes using repeat measures. So I'll explain this a little bit more on the next couple of slides. So by performing a study longitudinally as opposed to more cross-sectionally, we can actually improve our statistical power. So what may have taken, before an N of 20 animals can now take 4 times less. So the benefits here include lower intra-animal variability, so where every animal serves as its own control and we can use paired statistical tests. This can dramatically reduce the variability that we can see be caused by genetics or environment or various physiological changes between animals. And so that tends to be a source of noise, if we're considering a cross-sectional design. So in this case, you can use smaller sample sizes because the variability will be lower. So fewer animals are going to be needed to detect the statistically significant effect. And then, of course, you'll have higher sensitivity, and the longitudinal imaging allows you to track dynamic processes, as we discussed, within the same subject, meaning then that we get better temporal resolution as well. So now seeing this kind of in the context of Cintiqa's line of in vivo optical imaging systems, we have the Newton 7.0. So in this example here, the authors, they used a mouse model of acute lung injury to evaluate in vivo uptake and biodistribution of endothelium-derived extracellular vesicles, or EVs, and then those modified with lung-specific cell targeting peptides over the period of 2 days. So using DIR, the fluorescent signal of targeted peptide EVs had mainly accumulated there. You can see in the lung region at the 2 hour mark, which was statistically different from that of the EVs alone. And then about a day later, so 48 hours after the original injection, the signal had actually decreased, but the group still showed longer retention time in the lungs or the epigastric region compared to the other groups. So in this example, the statistical significance was able to be achieved only using an N of 3 for each group. Again, because those animals served as their own internal controls as opposed to having to sacrifice animals at each time point. So again, this highlights that the number of animals required can be vastly reduced using proper power calculations and repeat measurements. So once again, not only does this support better animal welfare, but it's also less labor intensive and less costly. So briefly, the Newton 7.0 systems, they're going to help you achieve 2D and 3D bioluminescence, the 2D fluorescence in the visible near 1 and at near 2 spectra, basically for all of your in vivo, ex vivo, and in vitro research applications. Not only is it going to be about choosing the right imaging system, so as you can see here, we have a variety of different models for the Newton, but it's also about choosing the right model for the question at hand. So this can sometimes start with a well-designed cell culture system or a refined transgenic animal model that might be later on a little bit more easier to adapt and scale. But these again are all considerations that need to be made upfront whenever you're designing your experiment because it can be even more costly and be less ethical to pursue down a line of experiments with animals that aren't the right mouse, perhaps using a certain wild type mouse that's not going to be as translatable in the end, and then again opting for something that's more humanized or genetically altered to begin with. So how does this all kind of fit together, like I said, with the Newton product line of optical imagers that Cintiqa offers? So again, if a cell culture experiment or a biologically relevant animal model is needed, how are we able to champion this with our Newton systems by way of replacing? So we can replace animal use with in vitro assays. So non-animal models can also be used. We see non-vertebrates and alternative organisms as well. Zebrafish, a variety of different insects. The adjustable field of view that the system employs and motorized focus abilities allows you to do petri dishes, 96 well plates, you name it. You're also able to or rather change the temperature of the pad so that if you don't need to have the heat turned on, you can turn it back down to room temperature. There's a lot of flexibility and versatility with these systems. Further, one last thing that I like to mention as well, whenever it comes to some of these alternative organisms and methods, Cintiqa is really great at offering application support. So if there's any kind of protocol refinement, you have some questions about different acquisition settings that you'd like to use, feel free to reach out to the Cintiqa crew and we can provide remote support for that. So whenever it comes to the reduced principle, smaller sample sizes can be used because the Newton systems are highly accurate and very efficient at data collection. So with having the widest lens on the market, you're able to pick up even the faintest signals, which again achieves or is able to offer really high sensitivity and specificity. With the deeply cooled cameras, it's helping to maximize the signal to noise ratio, allowing for really low background noise and picking up better specificity. So this all helps again to eliminate the chances of false negatives, false positives, which can creep up in other methods. Next, these all-in-one systems are capable of multiplexing up to 6 different channels all at once. And then you can also achieve volumetric insights as well with the 3D BLI tomography module, which is going to offer non-ionizing types of acquisitions, meaning that there's no need for, additional animal cohorts to be dedicated, perhaps specifically to CT, no special room requirements, etc. Finally, with the Newton's FT900 SWIR or near 2 capabilities, your studies won't be as impacted by the same common drawbacks that fluorescent imaging in the visible spectrum or the near 1 spectrum, where we see, challenges with light propagation in living tissues. So more specifically, near 2 light, it creates a lot less autofluorescence, thereby enhancing the signal to noise ratio. It's going to scatter less light through different biological tissues, so brain, skin, bone, muscle. There's also less absorption of this type of light through water, lipids, and other chromophores such as hemoglobin and melanin. So collectively, what this means is that, perhaps in order to achieve better depth of penetration and image deep tissues, you won't need to euthanize your animal and have a terminal endpoint where those are tissues that must be resected. In this case, you're actually able to do it non-invasively in vivo and then again longitudinally over time in order to still capture those same biological insights. So because the Newton systems are built with NIST traceable calibrated detectors, the light signals directly measured in physical units, so photons per second. This means that measurements are performed independent of the system's parameters and then are directly comparable across experiments. So this actually allows you to track the performance of your device over time, again, instilling confidence in your data and all of your collections, and then reducing the need for potential repeat experiments into the future. Importantly, this also makes, relative data comparisons to other in vivo optical imaging systems possible. The last point that I want to emphasize here as well on this slide is that because journals are starting to require more and more that data be made publicly available whenever you go to publish, scientists here benefit greatly from the Newton software being license free. So data sharing within and across labs globally is therefore streamlined and made easy. So taken together, time, cost, reliance on animal testing with the Newton is dramatically reduced. The last major thing that I want to highlight here then is the refine with the Newton system. So this platform is really designed with animal welfare in mind. Minimizing animal stress is of utmost importance, both ethically for the test subjects, but also because achieving accurate physiological data relies on creating low stress, consistent conditions. Otherwise, what can happen is the data is skewed, and then therefore, you have to repeat your experiments in order to get these more ideal low stress conditions. The next thing I want to mention is that with having, the wide lens aperture, we're able to collect the maximal amount of light all at once. This means that we'll have faster acquisition speeds, and then the animals therefore spend less time under anesthesia. So depending on the model that you select, there's quite a number of them, but the different Newton systems, you can scan, from 1 all the way up to 10 mice at once. So this reduces the amount of time that you have to spend swapping animals in and out of their home cages, which is also known to be, a contributor to having or instilling higher levels of stress for your animals. Next to Newton systems, they're also built with an animal platform that's user controlled for the temperature. So you can change the degrees from 20, so kind of more room temperature, up to about 37 degrees Celsius. The heated mouse platform also ensures that animals, they remain more comfortable. There's less major fluctuations in body or core body temperature, which can confound physiological responses. Another point to mention here is that we also offer a HEPA filtered isolation chamber as an add-on as well. So this can be used in studies that are requiring sterile conditions for any immunocompromised models. And then finally is our integrated anesthesia setup that features a red low stress induction chamber as well as a charcoal gas canister for active gas scavenging. So again, all of these components really kind of intersect to deliver the highest welfare possible. Um, I think the last thing that I just want to mention is really that when it comes to, doing your science and doing it well, not only is just the ethics of being kind to the animals that give back so much to us, having the data come out accurate really does translate to better outcomes and more human relevant information that we will see kind of translate to better benchside or bedside outcomes. I think in terms of the closing remarks, I just want to finish off by saying that as we step towards NAMS, we still have to stay committed to the three R's and the ethical use of animal testing. So, optical imaging, it offers a really powerful tool for studying complex systems. It enables researchers to refine their animal welfare practices, as well as getting better statistical power and accuracy in their data outcomes, reducing the number of animals overall. And of course, with proper study design and the strategic investment in the Noon platform, running in parallel to NAMS, you're able to really further the goal of the three R's and improve overall translational success and improve really human health span. Okay, so I think this was mentioned just briefly. I'll add this one here as well by Gabe during our polls. Essentially, over the next month, we've got a couple of really exciting conferences coming up. So, the Preclinical Imaging Consortium, we're going to be delivering a presentation featuring the Newton. We'll also have the FT100 Newton model on display at AACR there in San Diego. And so we really do invite you to tune into those presentations, come by our booth, have some awesome science discussions with my colleagues that will be present. And we'll be there to answer, yeah, all of your research questions. Okay, so I want to just save a little bit of time now if there are any questions in the Q&A or if there's anything that popped up in the chat throughout the course of the presentation. But otherwise, I just want to thank everyone for your time.
Okay, thank you, Jade. So we do have a couple pre-submitted questions and a couple live questions as well. So I'm going to
Sure.
start with the live questions for now. And the first one is by Anthony, and he says the following. You mentioned autofluorescence issues with bioluminescence imaging. Is this an issue with bioluminescence or just fluorescence imaging?
Yeah, sorry, great question. And if that's kind of how it came across, I apologize for misspeaking. So, no, you won't actually get any autofluorescence for bioluminescence imaging. It's one of the benefits of BLI is that really the only thing that is emitting light inside that chamber is the cells that would be expressing the luciferase. And so in that case, no, autofluorescence is going to be related more so to doing any kind of FLI applications in the visible or near one spectrum, really.
Thanks, Jade. We do have a following question from Anthony. He also mentioned, how do you minimize noise from the camera to ensure sensitivity?
Yeah, another really good question. So you can minimize noise by having lower acquisition times. So the longer that you have the camera or the exposure time set to, so let's say you're trying to collect an acquisition for something that is a really low emitting light source or signal, you might be tempted to plug in, 3 minutes for the exposure time, but you're going to end up picking up a lot more noise and background just because there's going to be electrons and heat coming off from the detector from the camera. So what I would suggest doing is actually changing the binning. For doing any kind of BLI applications, you'll likely already have the lens aperture fully open. So what's great about the Newton system is that it has the widest lens aperture that you can get. It's about an f-stop of 0.7, which is, I think, 40% more open than some of the other systems on the market. So that's one thing. And then I also mentioned binning. So with that, this is you've got a lot of flexibility going, 2 by 2, 4 by 4, all the way up to 16 by 16. And what it's doing is you're combining pixels to essentially capture more light. And so then with that, you'll see a higher sensitive or you'll see better sensitivity, but there's a bit of a trade-off with resolution and just kind of how the physics of all that work out. So while in some cases, having higher sensitivity is going to be more important than having a really highly crisp, high resolution image, because again, what really matters is being able to analyze or detect and analyze the light signal.
Awesome. Thank you so much. Um, we have another question from Jan. Jan, we're gonna follow up with you after the session just to give you your answer. And Alok, thank you. He says thank you. Thank you as well for joining the session. So we're gonna move on to the pre-submitted questions. Maybe just encourage everyone to ask another question, but for now, we're just gonna move on to these, as mentioned earlier. Yeah. So the next question is, when designing a study, what are the primary factors for choosing between bioluminescence and fluorescence, such as near to? And how does the system facilitate multimodal workflows with other imaging modalities?
Yeah. Okay. So before I jump into answer that one, the one question about the cost of the noon, I think it's worthwhile just to mention, and like Gabe said there, to follow up with us, and we'll be happy to provide quotes, is that it is a very affordable platform, especially in comparison to some of the other similar products on the market. But in the sense that, affordability compared to even, other types of imaging equipment, the Newton optical imagers are a really great value. Gabe, your question there about when to opt for doing bioluminescence, is that what the question was versus near to?
Yes.
I think that's what it was. Okay, yeah, sorry. I don't have the question up in front of me. But yeah, so there's a couple of different reasons maybe why you would choose bioluminescence over near two. We talked in the presentation a little bit about why near two has a lot of benefits versus traditional fluorescence. But in the case of comparing it to bioluminescence, maybe with BLI, you're interested in looking specifically at the activity of a certain promoter. So the luciferase is under the control of a certain promoter. So whenever it turns on, that's whenever you get the bioluminescent signal. So that can be one unique application where you might need to actually employ a BLI animal model. It's going to be probably, the most appropriate choice for doing extremely low emitting signals, as well as doing brain tumors in vivo where you're trying to get through the skull. Bioluminescence can be a good choice for doing just that. Early tumor detection as well. So because you get really good sensitivity with bioluminescence, if you are interested in characterizing, again, the pathological sequelae of tumor growth, um, it can be important to see, at the earliest time point when some of those growths can start to be detected, and BLI can be appropriate for that. There are some challenges with looking at BLI and tumors in the late stage of those animal models, whenever the tumor starts to become more necrotic, but that's could probably be another webinar entirely of its own. But for near two as well, again, we talked about the benefits of having less autofluorescence, less photon scattering through biological tissues. So what you're going to be able to do then is better deep tissue imaging, better real-time monitoring of biodistribution, different cardiovascular applications as well. Looking at one example that comes to mind is using, ICG, which has a longer emission tail into the near two spectrum, and that's going to bind to various parts of the cardiovascular system to really give you some beautiful images. In terms of combining the modalities as well, maybe you have whenever you're using the multiplexing, you could have the BLI allowing you to visualize the tumor, and then potentially you're going to tag on a fluorescent probe to, a nano particle or a nanotherapeutic that's going to be targeting the tumor. And in that case, you can multiplex both BLI and FLI to get more information about what's happening inside of your specimen. In terms of other modalities as well, the one thing that I think is probably worth mentioning of the different Newton models that you can choose from, we recently are excited to be now offering X-ray capabilities with the FT500 system. So it's called the FT500X. So again, that is going to give you the usual or the standard bioluminescence and fluorescent applications as well as X-ray technology as well. I think that's everything I want to mention there.
Yeah,
Gabe, go ahead.
thank you. Thank you so much. We have another question from Anthony, and I don't know, Jade, if you can see it through your Q&A.
Yeah.
Uh, I was
Perfect.
gonna, we can either address this after the session or just
Yeah.
privately. Up to you.
Yeah, so I mean, I'm fine to just give a couple of really general points. So with the Newton system, it offers essentially rivaled level of sensitivity and detection. I can share some data with you offline as well that speaks to that point. Having the widest lens on the market, which I mentioned earlier, helps to support having really high sensitivity as well to achieve that. Um, we also have a really strong resolution on these systems, and the reason why that's important, I'll use an application as an example. You know, if you're working with an animal model where you have a primary tumor and it is metastatic in nature, and you end up with multiple tumor sites around the body, having improved resolution capabilities can help you to discern between different tumor sites. And so with the Newton, that's something that is really easily achievable. And I didn't show any of the images that I'm thinking of in this presentation, but again, I'd be happy to share them after the presentation.
Okay, thank you so much, Jade. We're going to move on to the next question, which is the following. For researchers drafting grant proposals, what specific expertise or resources are available to help us articulate the transition to non-animal models and demonstrate a rigorous commitment to the 3 Rs through the use of optical imaging technology?
Yeah, it's a good question, and I think that we'll be seeing more and more of this, of course, because of the movement towards including NIMS and having, clear justification in your proposals on how your animal research supports the 3 Rs. Just in the fact that, to publish these days, it does require a lot of layers of different experiments kind of combined to, characterize what's happening physiologically and then diving into mechanism. You inevitably should be incorporating, different aspects of NAMs as well as the ethical principles of the 3 Rs for your animal research. And then in terms of the supports and resources, there's definitely courses that you can take, workshops that you can attend to educate yourself. I've got a couple of websites in mind, kind of focused on the 3 Rs, and talking about, what type of language that you can use, how to best go about your research to frame it so that, again, it is very centered around making animal welfare a priority. Again, I think whenever you're able to communicate that animal welfare and keeping low stress for your animals and properly powering your studies, that comes through both by way of, again, it being more ethical and also just in the accuracy of your data outcomes. And that, of course, will support translational success for you in the future. So, in terms of the supports, I have a couple of websites that I can share. And I also think that, again, at Syndica, because we offer a lot of technical and then application support, if there is language that you need, you're putting in a grant application for one of the Newton systems, we're happy to supply you with not only the sole source documentation that really highlights the value propositions of these systems, but also in how you can word those sections in your grants for highlighting why the Newton, why optical imaging. Again, speaking through the lens of the 3 Rs and through refined approaches and creating more human relevant assessments.
Awesome, Jade. So the next question is, for those of us in, oh, my apologies. For those of us managing limited lab space or relying on busy core facility staff, What are the actual physical installation requirements and specialized utilities? How steep is the learning curve for the lab staff or entry-level technicians to operate the system and manage the animal workflow?
Yeah. So not only, with these systems will you require lower number of animals, meaning you need less cages being kept in your vivariums, but the physical aspect of the system itself, including, let's say, the integrated anesthesia setup, so where you've got the induction chamber, the air pump, because it doesn't require a big oxygen tank, that helps to lower the space and installation requirements as well. Really, that portion of it is just going to take up a small benchtop space as well as the PC computer that it comes with. So again, just a normal standard computer and computer monitor that are hooked up to the system. Now it depends on which model you end up opting for. Some of them are benchtop models, some of them are floor models. They are quite modular, so they fit nicely into lab spaces, beside other pieces of equipment, beside lab benches or on lab benches. So in terms of doing the kind of working through the installation process with you, we're able to provide you with all of those dimensions so that if you need to prepare or move some old pieces of lab equipment around, it's perfectly easy for you to do that. The other thing that's probably worth noting is that, in comparison to some of the other imaging modalities or equipment that might require more infrastructure changes, or I'm thinking, radiation requirements or certifications, it doesn't pose some of the same stresses as X-ray devices will, CT. Same thing for, MRI or PET in comparison. So people tend to find if they're opting for optical that it's quite an easy system and modality to incorporate into their workflow. The question at the end there about a learning curve with the device, it's very straightforward, and actually the software is very user-friendly. So it's quite a sleek interface. The acquisition software and the analysis software are integrated with one another. We find that whenever we're doing the installation and the training for these pieces of equipment, it's really only a few hours that are required with users, and then we offer multiple sessions. So if it is a core facility, if it is just one lab, but there's, let's say 10 different trainees or students, we can split people up into smaller groups so that they have better time both with testing it out themselves, but listening to instructions. And there's an opportunity really to ask lots of good questions to build out and refine their understanding before we leave. So again, we come on site for a few days. We do the installation over the course of one or two, and then we have some real good dedicated time to performing the user training. But again, in terms of the learning curve, it's not very steep at all.
Okay, thank you so much, Jade. So we've reached the end of the webinar today, and I would like to thank you, Jade, for the excellent presentation and the viewers for attending this session. I hope the session has provided you with a clearer understanding of new approach methodologies, as well as an insight on how the Newton 7.0 optical imaging system supports the principles of the three R's. And as mentioned earlier, if you're going to be attending PIC and the ACR, please stop by our booth and have a chat with Dr. Merrill or any of the Syntica uh, employees. If you have any additional questions in the coming days or weeks regarding the modalities we discussed, please feel free to contact us at Syntica, and we will be more than happy to continue with the conversation. Thank you all for taking the time to join us today, and we look forward to seeing you at a future Syntica event. And have a great rest of your day.
Thank you.